There's no shortcuts. If you want to study the entire tree of life, you have to go to where it exists.
When it comes to data collection at this scale, the main driver for all of this is how can we get an accurate or as accurate as possible representation of the ground truth biology that exists on our planet. At the scale of the data that we're looking at, that needs to be done by hundreds of millions of CPU hours, with hundreds of millions and billions of annotations on the protein level, the DNA level, taxonomic information, biological information, contextual information, and so we're now orders of magnitudes bigger than all public data in sheer size.
I mean, I've never had a team like this before. I've never done science like this before, and it's very refreshing. And it's fast-paced, really fast.
So we're in Costa Rica, and we're on our way to Tirampina Reserve. Costa Rica, I think, covers around 0.03% of the Earth's surface, but yet has around 6% of the world's biodiversity. So it's a really, really incredible place for us to have access to and for us to build a collaboration with. Essentially, what we're doing is we're converting biological material to data. And specifically with our work, when you look at a spoonful of soil, there could be as many living things in that spoonful of soil as there are humans on the planet. So what you've got is thousands of different jigsaw puzzles that you can piece together with DNA sequencing.
Wildly, we've only just scratched the surface of what humans know about biodiversity. So Basecamp is here to solve that problem and to digitize nature and be able to actually know all of the solutions that have already evolved so that humanity can learn from those and create better products, better solutions out of those. So we're trying to build a bridge between biotechnology and biodiversity, which hasn't previously existed.
I first got interested in molecular biology in grade 11. I was in science class, and we were first learning about how DNA gets turned into RNA, and then RNA gets turned into protein. And once I understood it, that there's these little machines inside of our cells that are constantly turning DNA into RNA and RNA into protein, it literally blew my mind. I can still remember where I sat in class. And at that point, I was hooked, and I had to understand more about this. And I think one of the things I've really started to appreciate is that biology is hitting a data wall. And essentially what that means is that we've gone as far as we can with the data we have right now. If you look at all the different protein language models that are trained, they're all reaching a plateau where more compute is not actually making the models better. It's because they ran out of data.
The very likely reason for this is that all of these models are trained off of public databases, which fundamentally are lacking in the diversity that actually exists out in the natural world. And so by building this data asset that truly representatively depicts the diversity that exists out in nature, we're going to be able to prove that scaling laws also hold true in biology and that you can push performance up to the point where it's really transformational for biotechnology, for healthcare, and for so many different industries that are reliant on understanding biological systems.
Well, at our core, we see ourselves as a data company, but AI models are really just the best way we see of interrogating data and building a representation of that data. We want to see foundation models explode in biology that can understand not just one protein's function, but potentially any protein or any gene, any genome in any context, and it can do this generalized task. We don't have that in biology yet. And so for us to get that right, you have to have foundational data sets that cover every corner of the design space that exists.
terms of what we know about biology, we're probably 17 to 20 orders of magnitude away between what we know and what we don't know, right? I tried to do the math once to kind of have an image in my head. It's about 5 drops of water compared to the Atlantic Ocean. The Atlantic Ocean being that's what we don't know about life on Earth, right? And so all the paradigms, the rules, how we think about mechanisms and any definitions that we have in biology, they're only centered around those 5 drops, right? And so the question obviously is like, does that still hold true when you look outside those 5 drops? There's a lot that happens in between, from basically having a piece of soil all the way to a deep learning model or using these models for commercial applications. Where my team really starts off with is with everything that happens in silico. So once we've extracted the DNA and we've sequenced the DNA, that's really where all the computational workflows are starting.
It's an incredibly complex process to take this natural world data and then turn it into something that's machine readable for the models that we're building. And we've got great partners that help us do this. We have Microsoft and NVIDIA, who that expertise, that depth of expertise on scaling models on really large compute systems is the best in the world. And we bring that together with our expertise on scaling data. Humans are terrible at speaking DNA. There's a reason we're not speaking in the language of DNA. It's complicated. This molecule that's in every single cell of ours, meters of it in each cell, and it's got rules to this code. There are regions of it that produce a protein, and a protein performs a function in your body. And these genes are regulated at certain times, and they work together to perform functions. And so there's this inherent language around what a strand of DNA will let an organism do. And it's different in every organism, right? A bacteria performs very different functions to what you perform, to what a banana performs.
And so if we're going to start reprogramming these, there are 2 things we need really. One, we need data, right? We need examples of these things from nature. And 2, we need compute, basically, because we want to start understanding this language of how these proteins recognize their sites. And so if we can start understanding that language, then ideally we can start reprogramming novel proteins that will hit the sites we care about.
Sample temp 24.81, pH 4.98. Stand by for air temp.
Context in biology matters with every single application and every single tool that we want to use, right? For the more complex applications, if we want to push biology further, right? Leveraging this context can be really important because we don't always know all the interactions of what is important, what is not important. And so rather by having a more comprehensive view of not just all the sequences and all the genomes, but also how they have evolved, we have a much more holistic way of understanding this, in case some of these features are actually really important.
If we're talking about bacteria, the genomes that we're sequencing in our samples, their genomes are replicating at each division, and a division might be several hours for a given species. There is constantly a generation of novel genomes, and these can just ambiently change, but also they can change based on all of the different changes that happen in the environment. So maybe a new strain comes in and suddenly there's a pressure there from competition, or maybe all of a sudden it rains and that microenvironment is radically changed because the soil is now flooded with moisture.
To see a vast improvement in the complexity of the biological design problems that we can tackle, we have to undergo scaling laws, and we have to scale the intelligence of the models that we work with. We need scaled compute, scaled models and their parameters and architectures, and finally scaled data.
So from the outset, when we think about this huge knowledge gap that we're facing, we've set ourselves the challenge that we want to grow our database not by 10% every year, but by 10x every year. And that's obviously a huge engineering challenge that we've set ourselves.
We finally have the opportunity to start modeling evolution. And if you think about it, evolution is the most powerful force in biology for sure. It might even be on the planet. Essentially, what we're doing here is we're not just using our database to start identifying novel proteins we can use, but we're using the AI models we're building actually to help us start evolving those proteins to have the features we want without doing millions of variants.
And obviously that has a huge impact with respect to the data processing, storage, the orchestration of such pipelines. And when we think about how we're processing this data, there's obviously a lot of QC that needs to happen. We do bioinformatics assemblies, we do annotations, we label this data with biological and bioinformatics tools. And so when we grow this data exponentially, we've been using Azure for this quite a lot, leveraging AKS and the container registry to orchestrate this and to basically massively scale these annotation pipelines and be really flexible about this, right? Because we don't always have a guarantee as to when a new sample comes in, and sometimes it takes a couple of weeks to get a sample. Sometimes we have a dump of 100 samples coming in on the same day. And so one of the things that's been really important for us as well is to be really, really flexible about the timing, the scale, the orchestration of doing so. NVIDIA comes in at the next stage when it comes to actually utilizing these data sets for foundation model training. So we work with the BioNeMo team at NVIDIA and many researchers there to optimize existing architectures for our data. Our data is quite different to public data, so also there we have to think about some of the additions or changes that we would have to make to architectures, and then obviously using NVIDIA GPUs to train some of these models with.
The scale of data that we're dealing with is astronomical. And it's not easy dealing with all of this data in terms of the models that you train, the scale they need to operate at, the scale of the compute that we need to run. And we've got great partners that help us really accelerate that. And so on the scaled model side, Microsoft's Azure team, the people we work with there, the infrastructure that they've built there really helps us run as fast as we can. And similarly, on the compute side of things, we can operate as fast as possible with the accelerated GPUs that we work with NVIDIA on, the people, the expertise that they built around all of this in life sciences as well. This is really productive, the way that we can now start scaling these biological models in a way that we just couldn't before, and no company has been able to before.
We are taking a sample of this incredible spot just below one waterfall and above another waterfall in Costa Rica. There's an incredible need in industry to find gene editing proteins that are able to be programmed really efficiently or that can be safe for human therapeutics, or maybe ones that are much smaller enzymes so they can be delivered effectively in a medicine. So we're able to look into our database and see the diversity of sequences that encode for these functions. But for that, you need to know what you're looking for. It's a targeted search. And I think something that's really exciting and emerging with the advance of different AI tools and ML models is the ability to learn from this huge corpus of data to create tools that can help us to find new functions and design new proteins that have never been thought of before.
In reality, the team is much bigger than just the people that are at Basecamp Research, and our network of partners really enable this. So let's say that we are interested in a certain enzyme that we've realised comes from a certain habitat or ecoregion. We're able to pull that lever and ask them to be specific and collect samples from specific areas. And what that allows us to do is to have this incredible flywheel effect where we collect these samples, they can be analysed, and we can get feedback from the data science team and machine learning teams and the commercial team and feed that back into the sampling regimes moving forward.
The mission of the Global Research Team is to map the world, map all of biodiversity, and that's an incredibly ambitious aim. That is a scientific aim that rivals the Human Genome Project in terms of a great leap forward in science's understanding. And it would be incomprehensible to think about doing something like this as just Basecamp Research, a startup of around 40 people at the moment between London and Boston. And what makes it possible to conceive of this, and not just to conceive of it, but to be excited about it and running after it, is the fact that it's not just our team, but it's our global network of collaborators.
Biodiversity is not just about conservation. It is a source of innovation. For doing innovation, we need the partnership between the public and private companies or public and private institutions. Doing that is difficult in some countries. In Costa Rica, we have a divorce between some public institutions and private companies because it's difficult to talk or to speak the same language. To carry out studies that include the biodiversity and then we can move to the biotechnology, we need at least two components. First, we need to know the knowledge about the biodiversity of the country, all the environments that in CENIBIOT from SENAT, we know that. We have the knowledge about biodiversity. We have knowledge of the different physicochemical conditions in different environments. We have experience with all the environments in Costa Rica. But we need innovative tools that let us to discover new things. And I think that Basecamp Research has these tools.
The places that we work with need to see this benefit from us having been there in the short and long term. So in the short term, we enhance capacity with places that have a bioeconomy. We build labs, we share data back, we train scientists how to do this type of data collection, and we let them use that data for academic studies and whatever purposes they might want to. And in the longer term, if we see any commercial upside to the work that we do, we pass revenue back to these countries to show that there is value in this data.
The project between CENIBIOT from SENAT and Basecamp Research, for me, is a very good example that how we can work together. My dream is to see how the basic science is translated into applied science. Why not that we can obtain one new enzyme, a new medicine? Maybe we can discover it together. And maybe that can change the world, maybe that can change the country. And of course, the idea is that with this scientific knowledge and with this all the development of a new medicine, a new enzyme, maybe we can help to conservation strategies in the country. Maybe we can bring part of these benefits with this research to obtain benefits for Costa Rica.
Across the board, this is pioneering work by the world experts that we've brought together. And we've had academic publications in multiple different countries now studying their biodiversity in all sorts of different ways. All the while, it's very, very aligned with our mission, which is to have this global depth of understanding of biology, to train these new generation of models, get past these performance walls and start to generate new biological products.
At its heart, Basecamp Research as a company is built on this idea that biology has the answers, and the process of evolution has led to this really truly remarkable complex system that shouldn't work, and yet it does. I think there's an incredible amount of inspiration that you can derive from the natural world, and being out in the field and being able to see the richness of life and be struck by how much complexity is there and how much there is to learn from is really important. Fundamentally, we're so reliant on biodiversity, and being able to study it and understand it is one of the most important things that you can do, I think.
I really believe having better foundational models is going to help in many different ways. New binders, new small molecules, new everything. I think we are living in the most exciting time in human history because I don't know what's going to happen next. And I don't think anyone does know because the rate of change is happening so fast. And now I get a front row seat at it. And it's great.
Biology touches not just one industry. We always talk about biotech, but life sciences is beyond biotech. It's the food we eat, it's the materials that we wear, and it's the medicines that we take. The future for Basecamp is to continue pushing on all of these fronts, to continue to scale the data at our core, but to realize that value through the new biology that comes out of that and get that into the hands of the pharma companies, the agricultural companies, the chemical companies who can really turn that into products that affect you and me, that consumers use and use in their everyday lives. All of these amazing things that we want in the world come down to our ability to control biology and design biology, and computers are how we do that. And fundamentally, computers need to see data, and they need to see scaled data. That's where Basecamp comes in. We solve that data wall problem so that we can then see these totally new products in biology start to be possible.